Great. All right, everybody, welcome back. Thank you for accommodating the scheduling change at the last minute. So I'm still Andrea, and I'm still happy to be here at the Russell Sage Foundation Summer Institute. We're going to pick up where we left off yesterday, talking about human genetics. I promise we're going to get more into the human side of things today. So this lecture is not very creatively titled Human Genetics 2. We're just going to quickly review Mendelian inheritance, and then we're going to dive straight into talking about association analysis as a paradigm shift in the field and how that was both very important in enabling modern day genomics and genetics, but also in changing the way that we think about the discipline and changing the way that others can possibly see the discipline. Then we'll get into a little bit of what functional follow-up looks like on the biology side. This is maybe a caveat to mention here is that this is absolutely from the point of view of somebody who works in drug development, and so we're very interested in the nitty-gritty details of how different molecules relate to each other. And then just a quick introduction to rare variant methods, since that is also super relevant here. So last time we talked about Mendelian inheritance. We had a lot of pedigrees that looked like this with circles and boxes, and they were shaded or not. We talked about putting allele calls on the boxes and being able to trace patterns in this way. We had an example about linkage analysis, where we looked at a bunch of examples in plants and in animals. So this is the fruit fly example from yesterday. This is the back cross, where we take the heterozygote individuals for two traits and the homozygous recessive individuals for two traits, cross them together, and then look at how the traits segregate together. We talked about how you can parse this out and you can ask, is the observed count different from the expected count? I should mention Mendel actually did this with peas, and the traits were smooth and wrinkled and yellow and green, and he found that they were not segregating independently, I believe. So this is all the stuff from yesterday. This is the way people are thinking about genetics being very simplistic, very deterministic, and really doing only a couple of traits and only a couple of markers at a time. So as a paradigm shift, linkage analysis assumed a lot of simple stuff. I basically just said that. And association analysis relaxes these assumptions on inheritance of patterns, doesn't make any assumptions on the number of loci involved, and just requires the usual kind of case control or quantitative trait data, and then lets us explore genetic markers in that context. So it's maybe worth it to start talking about the spectrum of allele frequency here. This is absolutely a continuum and not an either or sort of situation, but in general, we do talk about common versus rare variants quite a bit in the field. So Mendelian disorders are really rare. So consequently, Mendelian disease variants are also rare. Linkage analysis can require many large pedigrees. We talked about that yesterday, about like you wouldn't necessarily have the power to make inference from a single pedigree. Biological insight can be super limited in the linkage case. And GWAS is just a totally different approach that allows us to look at each marker one at a time on unrelated samples and draw conclusions. And then there's a lot of power in just the ability to do that. So genotype arrays popularized GWAS, providing the genotypes at many loci simultaneously. This is some of the data that you're going to be working with is from genotype arrays. And we'll talk a little bit more in depth after lunch about how the genotype arrays actually work. But as a quick introduction, this is a technology allowing investigators to query many biallelic SNPs at once. This biallelic piece is a small but important detail. So it means that I only have two options at this site. You can be C or you can be T, but being G is not an option. In 2000, AFI had an array with almost 1,500 SNPs on it. By 2009, arrays were advertising over 1 million SNPs. And so this is fairly quick development. Today, Affy and Illumina both offer commercial SNP arrays with over 4 million SNPs. And just to give you an idea of how quickly the cost of this has leveled out, and I actually had to update this from the last time we even had this meeting. So in 2000, SNP arrays were well over $1,000, depending on what you were trying to do, depending on how custom it was. By 2009, we had gone up to 1 million SNPs, and these were going for about $700 a piece. In 2019, the last time this meeting was held, Affy and Illumina were both around the 150 range for 4 million SNPs, and today those same chips are still dropping in price. Imagine that you could get this down to something like $30 if you ordered enough of them. But even a single array is being quoted at under $70. And so this just incredible decrease in price is also something that's driving this technology as a really important tool. So GWAS, as I'm sure you all know, stands for Genome-Wide Association Study. And for each SNP, one at a time, we're running a simple regression model. And this is certainly oversimplified because I don't have all my covariates in here. This is just for sort of completeness, that for quantitative traits, you would run kind of the standard ordinary least squares regression model. And for dichotomous traits, you would do a standard logistic regression. And this was kind of the high-level thinking on how we would use something like a SNP chip to answer questions about people's phenotypes. So this was the sort of standard notation a very long time ago, was that it kind of converged with the standard regression notation where you'd have a phenotype and a genotype. You would estimate this coefficient, and you would ask, is this coefficient different from 0? So positive effects and negative effects are allowed. This is just some simulated data that I made for this talk. So when you make a Manhattan plot, you have your genomic position on the bottom, and people normally just stack the chromosomes, even though this end of chromosome 10 and this end of chromosome 11 have nothing to do with each other. And then you would put your negative log p values on the y-axis. And each one of these plots, each one of these points would represent a SNP. And the idea being that if you looked for your SNPs that stuck out, you would then dive down deeper into those locations. So I think we're going to walk through that. So the standard kind of conventional GWAS significant threshold is 5 times 10 to the minus 8. This is a compromise between a Bonferroni correction and the multiple tests. For multiple tests and no correction, and this has to do with how correlated the SNPs are. And so there was a big debate about where this threshold should be when folks first started doing GWAS, but as a field, we've sort of compromised and decided that this is around the right level of correction, given that so many of the SNPs are correlated with one another. And yeah, go ahead.
Was that people-driven or just kind of like a debate?
So there were folks that were advocating for Bonferroni correction initially, and that was obviously empirically driven. But even at the time, sample sizes, you couldn't get your sample size for any kind of complex trait to be something that was reasonable so that you could start applying a Bonferroni correction, and especially as we're going from a couple hundred SNPs to millions of SNPs, that correction gets a lot steeper, a lot faster than we think we wanted to be correcting. And so then there was an empirically driven effort to talk about effectively how many SNPs are there and what does the correlation structure look like. And that debate, in my understanding, is what put this debate to rest and allowed us to keep our sort of conventional threshold. Dan?
Just to add to that, we've talked about the frequentist and the Bayesian justification for genomic significance, and actually both of those were at play in the discussion. So there was on the frequentist side there was this estimation of the number of effective tests that you're doing, and then on the Bayesian side there was discussion of prior probabilities and exactly the sort of thing we just talked about. We sort of converged on this.
So yesterday we talked about LD and linkage, and what folks started noticing was that if you had peaks in your GWAS, if you had a Manhattan plot like this one, where there was one point just sticking out all by itself, sometimes those were artifacts. In fact, very often they were artifacts. And as the genotyping got denser and denser, folks figured out that if you weren't seeing some evidence of LD, this correlation between SNPs, that something might be wrong and that you might want to quality check that particular SNP. So many factors can influence GWAS results. Obviously, poor data quality, garbage in, garbage out is a concept that we're all familiar with. And that can be in SNPs or in samples. And so there are two dimensions to the data matrix, and so it's important to think about quality on both of them. And then population stratification, which we've already done a great deal of discussion about. So this is, you will have this information more formally a little bit later, but this is just an outline of the steps in filtering SNPs. So SNP call quality, again, after lunch we'll talk about exactly how the SNPs get called, but sometimes the result isn't clear. And so when you get a quality score assigned with your SNPs, you want to take that into account. SNP genotyping rate, so if you're missing a lot of SNPs, or if you're missing a lot of calls on a particular locus, it might tell you something about how well that probe works. Duplicate sample concordance, so if you happen to have run the same sample more than once in your study, you would want to make sure that these results line up. And if they don't, then maybe there's something wrong on the SNP side. In terms of minor allele frequency, if things are very, very rare, they might not be helping you out a whole lot. That depends on what you're looking for on the study design. And then Hardy-Weinberg equilibrium, which I'm going to take a second to talk about now. So this idea is that if allele A has a frequency P and the allele B has a frequency Q, which is just 1 minus P, that the frequency of all of these genotypes should meet their expectations given me. So this has nothing to do with a second marker. This is just one single marker. And the idea is, this is getting at the idea of whether or not this particular marker is assorting randomly in the population. If it's subject to some kind of selection where you get homozygous here and homozygous there, but no heterozygotes, then that might suggest that the SNP doesn't meet some of our assumptions further down the pipeline. I should also mention that this can be real. There are things that fall out of Hardy-Weinberg equilibrium, but often when this happens, it can also indicate an artifact of calling on the chip. So it's often that the chips have trouble calling the heads, is my understanding. So we also have to filter samples. You want to check that the chromosomal sex matches the reported sex. That one's really nice and easy, whether or not an individual has a Y chromosome or not. The genotyping call rate, so if you're missing a whole bunch of markers, maybe something about that sample wasn't quite right, it wasn't run properly, and then maybe the sample might want to be disregarded. You want to check for duplicate samples. Often these things are run in labs, and people sometimes make mistakes. And so we've definitely had moments where we've caught tube swaps or having the same sample in a data set where we didn't expect to. And as you might imagine, if you match every single SNP at every single locus, chances are that's a duplicate and not a twin somebody doesn't know about. So speaking of relatedness, you also want to check and see if you have any family members that either didn't know that they were all entering the same study, or maybe didn't report that completely accurately in your study design, and then also control for population structure, which we've already talked about quite a bit. So the way this, in very broad strokes, the way this normally works is you would filter out all of these samples by a simple pass-fail criteria. You would say either they pass or they fail, and then you would remove the ones that fail, and then adjusting for PCA is the way most folks control for population structure. Yeah?
Excuse me, you said the SNP genotyping rate should be above 90%. Does that mean that 90% of the people in our sample should be genotyped for that SNP? Otherwise, we throw out we remove the SNP?
On the back,
Just like
here, yeah. Yeah, so that's per SNP. So that's saying of all the samples that you've run, duplicate or not, do you get it to 90%? And depending on what you're doing and depending on the study design and what you're willing to tolerate, this wiggles around a little bit.
Okay, so per SNP, 90% of the people should be genotyped for that SNP?
Yes.
Otherwise,
Otherwise, you remove the SNP,
the SNP, yeah.
not the sample.
And what is call quality?
Sorry?
Call quality. First.
Call quality. Okay, so every SNP call in the raw data will come with a quality score. And usually there's a threshold, again, depending on what you're doing and on which technology and which array you're using.
Is it meant to as a measure of how confident you are in the calls?
Yeah.
What is it called?
What is the call? We're actually going to talk about how they're called after lunch in great detail, but it's a ratio of fluorescence. So
it's about what what a person's genotype is. So you call them as having AA. Or
Oh, okay. So the genotype.
oh yeah, how and as the algorithm does that, they call that a call. I've called this GG. I've called this TT. Right. Sorry, little piece of jargon. Cool. All right, so population structure. This is more for completeness than anything since we've talked an awful lot about population structure already. GWAS is ultimately an association test of allele frequency versus the trait of interest. And so individuals from different populations have lots of different alleles that exist at different frequencies. We already know that. And so within continental populations, there are gradients of minor allele frequencies that are quite common. This is most first demonstrated in European populations. But the idea being that this structure can confound your GWAS analysis. And the more of these variants we have, the greater the risk of that becomes. And that's where controlling for population structure becomes very important. So on the first day, we talked about this idea that we are all descended from a common ancestor, and that we have migrated all over the world and evolved into several distinct subpopulations. And so principal components is a way that we deal with that. So this was invented in 1901 by Carl Pearson, and it aims to convert a set of possibly correlated observations to a set of linearly uncorrelated variables. This is a while before GWAS came on the scene. So PC1 is going to capture the maximum amount of variation in a sample in a single vector. PC2, the same, but this is conditional on it being orthogonal to PC1. So I know we can only imagine things in 3 space, but if you think about cloud of points, the line that best fits that data would be PC1. The PC2 is the next line that captures kind of the most data coming, and so on and so forth, even though it is very difficult to start imagining orthogonal vectors beyond 2 in 3 space. So this is super early, so this is 2005 already, early for the GWAS days. So this is the HapMap sample populations. CEU was Caucasian individuals from Utah. HCB was Han Chinese. JPT was Japanese samples collected in Tokyo. And URI is a Yoruban cohort. And so you can see if we just look at PC1, if we look at PC1 and PC2, what you end up with is the Yoruban individuals over here, the CEU individuals over there, and then the Japanese and Chinese cohorts at a third cluster. HapMap then expanded. This was an international consortium dedicated to capturing this kind of variation. And so you can see that we've now added a number of populations. It should have been a trivia question to identify all the abbreviations. I think I know most of them, but there's probably a couple I don't know. So you can see we're adding bottlenecked and very specific populations like Finns, and then there are other more historically admixed populations. I think this is Mexicans from Mexico City. And so you can start to see that this picture starts to flesh out a little bit. Here we still have CEU is the dark blue circles, and so you can identify them here. And then the sort of African cluster starting to be here, and then Asia over here. So as more and more data were collected, you can start to make really cool graphics. PCA captures information on continental ancestry, but it also gives you some idea of the continuous population structure within a group. And so this is all of the European populations. It's standard practice, or at least it was, the last time I did a GWAS, to include PCs 1 through 10 as covariates. But if we just look at the first two in Europe, you can almost recapitulate this map by looking at where folks fall in PCA score. And this was a John November paper released in 2008. And this is a cool result because it gave us this idea that there are actually things that have some kind of decline in allele frequency, that it is more common over here, less common over here, and then middling in the middle. Okay. So fast forward to 2009. This is Stefan Ripke's 2009 paper, which is the Schizophrenia GWAS, this great big hit right in the middle of the MHC region. And so this was, oh yeah.
Yeah, so coming from the world of psychology, I'm always kind of wary about controlling for a ton of variables.
Yeah.
Because you might accidentally control for the thing you want to keep in
Yep.
the model. Is there evidence that, like, I know that, like, when you control for 10 principal components, you generally get rid of the population stratification, but are there other things you're accidentally controlling for that you want to keep in the model?
So kind of the classical overfitting question.
Yeah, yeah.
My understanding is that not usually. And if something is super associated with, like even if you're associated with ancestry, like say one of these weird Klein ones, you should still get a significant result even if you've adjusted for the PC, because you're not adjusting for individual SNPs, right? You're just adjusting for like, so if you look at PC1 for all your samples, it's just a value. And it's not going to, unless you get really far down the scale, it's not going to correspond one-to-one with like any particular SNP. Yeah.
You need it to, because these PCs are going to capture genome-wide sorts of phenomena, right? Mm-hmm. And so the thing that would be getting rid of would have to be something that's also just like spread across the whole genome even more than what Ancestry does. Okay. And so I mean, I guess if you imagine there's something like incredibly polygenic, then you know, then maybe, but in practice, I think you'd have to get to pretty high order PCs before you'd start worrying about losing signal. But I mean, I don't think there's empirical work on this, but just kind of thinking through what the PCs would actually capture, it seems like it's unlikely that you'd lose anything really, really meaningful. PCs also sometimes actually can capture not just ancestry, but like technical artifacts. So like if some of your sample was genotyped on one chip and another was genotyped on another chip, because you know these are again kind of genome-wide things that would be aggregated across a lot of SNPs. So it's not just stratification that PCA can help adjust for.
I think, Dan?
I think it's useful. I think the main case to think about for the potential for over-controlling that's useful to think about is if you had two ancestral groups, let's say one of them was homozygous for like no, you know, it was homozygous for the minor allele for allele A, and the other group is homozygous for allele B, and there is an effect of that gene, or that variant, then you're losing the ability to see that difference. All the variation is coming from those two ancestry, that cross-ancestry comparisons. So controlling for the PC, you lose all the signal. But you know, it's not clear you want to identify for that cross-ancestry difference, because it could be from this population stratification confounding. And more realistically, there's going to be variation within each population. So you may be losing a lot of power if most of the variation is between ancestry group, but as long as there's variation within population, you won't lose the ability to see this. You won't actually lose the signal.
Cool, thanks.
Cool. I'm also realizing that I'm spending a lot of time facing this way, so if you are in this half of the room and you want to get my attention, just shout. Okay.
Just shout.
or I could see you now.
So my question is regarding like the level of stratification.
Yeah.
I'm assuming so the highest might be like, okay, different ancestors, or within a given ancestry will also have like stratification. So is it like a GWAS has to be done within one ancestry?
Gotcha. So no, the GWAS doesn't have to be done within one ancestry group, and that's one of the cool things that PCA allows us to do. And it also buys you correction within an ancestry group for kind of common ancestors, which is the real power of this. There was a method actually prior to PCA called genomic control that forced you to specify how many subpopulations that you thought you had, and that was a major downside, that you could really only control in this categorical way. So I think that's the thing that people really like about PCA.
So let's say like you have like a smaller number of people come from a certain ancestry.
Mm-hmm.
Would you claim like genome-wide level of significance like for an ancestry that has a smaller number of?
I see. It depends on what you're trying to get significance with association to. So in general, we're not trying to associate things to ancestry, right? We're trying to associate them to some phenotype. So you could have folks with different ancestry in your GWAS and still use those samples and have a valid GWAS at the end. If you wanted to compare your results across ancestry groups, that's where you would get into trouble. So you need enough sample size to actually run that analysis on your ancestry groups.
It's all really, like, you know, it's all quite messy generally. I don't think we totally understand the degree to which stratification is causing us problems. We know that it's causing us problems because we can look to see. It's easy to test if there's stratification, and we see that there's stratification. And we can control for it. Even doing that, it looks like there's still residual stratification. And so the question is, like, for the application that you're doing, like, have you gotten rid of enough that you can learn useful, interesting, replicable things? But it's all you know. I don't. The jury's still out on a lot of these kinds of things. We're still learning about this.
Yeah.
My understanding is that dimensionality reduction in a lot of different applications of biology is really important, and in some of those applications, people have been moving away from PCA.
Mm-hmm.
Is there a reason, like, is PCA particularly well set up to identify these different ancestries in a way that it's not like when you're working with cells?
Gotcha. Are you talking about the sort of blanking on the name of it for expression data? There's
Like UMAP or?
Like UMAP and TSNE and that kind of family
I
stuff.
think TSNE is like, my understanding is that it's just sort of like a visualization tool that is the same true for UMAP?
I get the impression that UMAP doesn't have the same
Okay.
set of, Brayman's nodding, which makes me think yes it is.
Yeah, so it's the question of dimensionality reduction that's preserving all of the variance on your chosen set of dimensions versus prioritizing like the maximum amount you can squeeze into a visualization or whatever, as opposed to like maintaining orthogonality between things. There are theoretical arguments from some simplistic models of like population drift that would suggest that PCA is the optimal choice. That is relative to those simplistic choices, and even then, PCA is not a perfect solution.
Cool.
It does pretty well for long-run stratification. I think when you get to kind of more recent migration and things, there's just not big enough differences between groups. You know, that's where it starts kind of failing, I think.
Yeah.
There's some sample size dependence there, but essentially you need power to pick up on whatever the necessary axis of that differentiation that happens to matter to your phenotype. So as with all things, your power to pick up that axis is simple size dependent.
Does that help?
Yeah, thanks.
All right, so if we go back to our simple Manhattan plot from the schizophrenia GWAS, and we look at one of these loci just super zoomed in, you can see this little triangle is the thing that we saw on the previous plot, but this is just the top hit. And there are actually a whole bunch of points here, sitting here in a cloud, and this is due to LD, like we talked about yesterday. And so the idea here is that one of these is, at least one of these, is hopefully truly associated with the phenotype that we're interested in, which in this case is schizophrenia. But we don't necessarily know that it's this top one. This is just the one with the highest p-value. So should also mention that not all of these SNPs were actually typed on the array. A lot of them were, but some of them were not. And so some of these were imputed. And I'm going to talk about how imputation works kind of on an intuitive level and not get into the nitty-gritty of the math. So SNP arrays provide information on some number of genomic locations. That number of locations is increasing, as we talked about. And we can infer missing SNP genotypes from known haplotypes. I was a super baby grad student when this came out, and it's maybe worth mentioning there were a lot of naysayers about this. People were saying, like, you can't do that, this is never going to work, you're just making up data, and it turns out it does work. So maybe something to keep in mind. So in HapMap, and now 1,000 genomes, they provide large, dense sets of haplotypes because we have sequencing data on all of those people. So say we have these SNPs genotyped in a region. This is just a cartoon. And we have these known haplotypes where maybe there's a really polymorphic SNP right here, but everything else is more or less similar. Then we can fill in the bases, assuming that this person has the blue haplotype and this person has the green haplotype. This is kind of intuitively how imputation is working. And you can imagine that this gets much more sophisticated and much more accurate as you get more and more haplotypes to draw from and as your markers get closer and closer together. So the UK Biobank imputing into 1,000 genomes and resulted in cleaned GWAS data that has 10.8 million variants, which is a lot more than was typed. Imputation usually happens in 2 steps. So there's a haplotype estimation step, and this happens via hidden Markov model. This can take a while. And then finding the closest reference haplotypes is generally the 2 pieces that this works. I have not kept up with the literature, but last I was running this kind of thing, Mock, Minimac, Impute2, and Vivo were all kind of front runners. Yeah.
In the previous slide,
Yeah.
in the top right, there are some base pairs with like a stacked
Yeah.
nucleotide. In sequencing data, would that just show up as like a low quality imputation?
In sequencing data, okay, so maybe this allele probably wouldn't be imputed.
Okay.
This is supposed to be the representation of a haplotype. And often when you see the letters stacked like this, it means it is supposed to convey frequency.
I see.
So usually the top one is going to be the most frequent, and then the bottom ones are less frequent. There's a name for that kind of logo where you see the 4 bases and they have relative sizes relative to their frequency. Yeah.
So would would they be called differently in terms of SNE, like using these?
So similar to with base pair calls, you get a quality score with your imputation. So if this really was like a half and half SNP on this haplotype and all you had was TTT, you might not call this SNP at all. Like you might not use it, or it might not even be in your imputation panel. It kind of depends on, because the imputation panels don't tend to include things that you then have to say, I don't know about immediately after. Does that help?
Yeah.
Sweet. Yeah.
Am I correct in my understanding that computation is not natural ancestry, it's just general ancestry?
Right. And the idea being that for your imputation panel, you need a representative sample of the ancestries that you have in your study. And that's why things like 1,000 genomes and HapMap become really useful in that we're mapping to haplotypes that are hopefully at least from the same populations. Cool? Yeah.
I'm just curious, what causes the missingness?
Missingness. Here?
Yeah.
Okay, so this is, what causes it is that we didn't design a probe on our chip array to ask about these bases. We're only asking about these bases specifically. Does that address your question, or did I misunderstand it? Okay, cool. So there's a lot of ways to do imputation. Imputation can be quite computationally intensive, and so most of the progress being made in this field has to do with computational speed. Accuracy, of course, is always being improved, but speed is the big one. So common versus rare variants in GWAS. Each SNP is assumed to act additively, whether or not that's true. I'm sure you guys have done some discussion of additive versus dominance models, and will continue to. GWAS leverages statistical power of common variation across many, many samples. And so more samples equals more power, which we all know from our tote bags. Many GWAS significant loci represent loci of small effect on the trait. And this is pretty important for complex trait genetics. And then power in GWAS is reduced for low frequency variants. So in this case, they were setting the threshold for being a low frequency variant at less than 0.01%. So this is from a 2010 Nature Reviews genetics paper that kind of talks about this relationship of this trade-off between allele frequency, which is shown on the x-axis. So this is stuff that would be very, very rare, going to more common things over here, and then the effect size that you're likely to see. And so you have low, modest, intermediate, and high, and going up and up and up. And so this hypothesis was sort of formed that rare alleles causing Mendelian disease would live over here. They would be very rare, and they would have a really big effect. And then common variants that are implemented in common disease or not implemented in, or not implicated in anything at all would live over here. And so there are a few examples on the off diagonals, but these do tend to be quite rare. So high effect common variants and rare variants of small effect. So these guys would be really hard to identify, right? If they're super rare and they have very small effect size, you need a whole lot of people to even measure this with any degree of power. And then, of course, there's lots of stuff in the middle. So fine mapping is what comes after you make this picture. And this is a whole bunch of SNPs are associated. Which ones do we follow up on? So GWAS peaks can be wide. They include no genes. And then you want to ask, what does this change or why? One gene. Then the obvious question to ask is, have we implicated this gene, or is it just a proximal relationship? More than one gene, and then you have the problem of deciding which gene you think is implicated. So determining a set of truly risk-conferring causal genes or SNPs from genotypes is an active area of research, and there are a lot of other places where this comes into play. I should also mention that this kind of definition of causality, I'm coming at this as a drug hunter, and so we're very interested in how different molecules interact with one another. And so we're really looking for a literal explanation for this. It's also worth noting that sometimes LD buddies, which is what my dissertation advisor Yoon Lee used to call them, things that are so close in LD that they always travel together, and there's not a whole lot you can do with your GWAS data because they will always give you the same information. That would be a case where LD is 1. So there are a lot of ways to follow up on your GWAS hits. using functional data. Some of them that stay more in the omics sort of space would be EQTLs, so expression quantitative locus. So we're looking for an association between the expression level of a transcript and a SNP. And I'll show you what that would look like in a second. A protein QTL, these start to get very expensive as measuring protein is more difficult. But that's the same idea that if you measure levels of protein and you associate that to the genotype, is there some kind of meaningful relationship? Then chromatin state and epigenetics and pathway analysis. This is computational, rapidly evolving, and super dependent on known annotations and how that data is sort of arranged. So these almost always require the tissue of interest. So if you're interested in something going on in the brain, you may or may not get the answers that you're looking for if you start doing measurements in blood. They require the time point of interest. So if you're interested in something that happens in early adolescence, patients in their mid-30s might not give you the answer you're looking for. And they are rarely enough to demonstrate causation by themselves, but taken together, you can start telling this story. So yesterday, we talked about the central dogma of biology, this RNA to DNA to protein idea. And so this first one, we're going to talk about EQTLs, is going to link DNA to RNA, showing that there's some first step there. And so supposing I have a bunch of SNPs, none of them is in a coding region, I might start constructing plots like this where I have a quantification of RNA, or if we're doing a protein QTL protein, and I'm looking for a linear trend among the genotypes. And so this one would be potentially a significant result. This one would be a null result. And so for the kinds of phenotypes we're often looking at in biology, genetic variation can be, oh, yeah, Dan.
Just to clarify, that data is coming from a different data set where
Yes.
we have different genotypes and measure the expression level.
Yeah, so these would have to be the same samples. So you would have to have expression data in addition to your, so the SNPs could be from the GWAS. If you also had expression data that you're interested in. Very few folks are that lucky, but there's no rule against using the GWAS data. Yeah.
In this context, we might be interested in things where the brain is the target tissue. Is this realistic that people are donating brain samples?
I think that debate is still ongoing. So a lot of folks will say, well, I can't get you brain biopsy, but we can grow NGN neurons in a dish.
Got it.
And then the question is, well, is that good enough? Is that close enough? Some people would say yes, some people would say no. And it depends on what you're looking at.
There's this brain scan data as well
Mhm.
from people who died and brain biopsy.
Yep. Yeah, there's brain scan. GTEx has a huge brain subset of samples as well, complete with regional annotation. There are folks in the world, mostly folks who are huge fans of single cell kind of sequencing, that would say that even those aren't enough because the people are dead, because they're not, obviously dying has a huge impact on your biology, and especially in the brain. And so that you're still not getting at what you might be interested in. And there are folks who think that it might be very specific to a cell type.
I see. So a lot of this existing data is from bulk.
Yeah, a lot of this is bulk RNA-seq. Single-cell RNA-seq data sets are starting to come online. This gets a little bit harder, especially a lot of brain-expressed stuff, at least that's been interesting in psychiatry, tends to be really lowly expressed. And then if you go to a single-cell paradigm, it's even more lowly expressed because you're trying to sequence a single cell. And so it's always that trade-off of like what information would we want in a perfect world versus what information is available and would actually give us the power to make a conclusion. So yeah, this gets really messy. Cool. And the idea here being like if you have some kind of genetic association, you have a phenotype that you're interested in, and you have a nominal statistical association here, what pieces in the middle can you add to the story to kind of bolster your argument? I would say even in pharmaceutical drug development, people don't necessarily hit every possible experiment, but you want to construct an array of these so that you can tell your story and it all kind of lines up and tells you the same thing. And the more of these connections that you can add, the stronger that your case is, and the more experiments you can think to do and to design more specifically, because a lot of things you can do one at a time relatively quickly and cheaply, but maybe not in parallel for all 108 of your GWAS hits. So a QTL, quantitative trait locus, just takes a continuous outcome, and that can be expression, that can be protein. There are even other assays that give you a quantitative readout that you can use for this. And so if you can show this way, you've shown causality, but you could also land yourself in the pleiotropy or linkage situation. And so often follow-up experiments, if you have, say, an EQTL and a SNP that's associated, is ruling out these other cases to see if you actually do indeed have causality. Yeah.
So, can you just define what a QTL is exactly? I see it all the time, and I don't know how I would define it. Like, what is it? What is
Gotcha.
a QTL?
QTL is a SNP association with a quantitative variable. So it's a demonstrated association. And then whatever's in front of the QTL would be the association with what?
Okay, so QTL could just be like a GWAS result if you're just saying just the QTL by itself, but then EQTL would be an association between the SNP and expression. Is that?
Yeah, I guess if you did a GWAS and your outcome in the GWAS was some kind of quantitative measurement, I guess you could call it a QTL. I think people normally use more molecular measurements, but
Okay.
and even years ago, like lots of folks were worried about splice QTLs. Like, can you look at how frequently a certain splice site occurs based on the genotype? You could imagine that if you had a SNP sitting right in that splice recognition site, that could change it a lot. But even upstream and downstream SNPs seem to have an effect, and some of them are more subtle. Yeah.
For SNPs that aren't near any genes, how do we know which genes to check?
We don't. It's a huge problem.
Yeah. I mean, I guess this is kind of a conversation about like generally when a SNP is kind of in like a gene desert, but it's doing something in the GWAS maybe. It's kind of a hard question to say what.
It's a super hard question, certainly just from the GWAS data.
Right.
So if you start pulling in supplementary data, you could start asking questions about expression QTLs, and there have been documented cases of QTLs spanning really, really long distances. So it's kind of trans EQTL kind of idea. And so you might get lucky and find one of those. But again, if you don't, it's very difficult to interpret as a result.
Yeah. And I sometimes have the bad habit of using the word SNP and the word gene interchangeably, but
Gotcha.
you know, it's I guess probably pretty rarely the case where it's obvious what SNP is associated with what gene.
Yeah, I mean, if it's in a coding region,
Sure,
that's nice and obvious. You get, I think the modern analysis tools call them tiers. So like if you take all of your GWAS hits, you would say that the tier 1 hits are immediately adjacent. They're in some bubble, it's often 10 kb in either direction, of one gene and only one gene. And then you might say, well, the tier 2 hits, there's 2 genes landing in that bubble. So now we just have to differentiate between, is it this gene or is it that gene? And then sometimes it's almost the opposite problem. It can also be quite difficult as well, as that you have something in a very gene-rich area. And then you just don't know what to do with the 25 genes that you've now uncovered. Okay. we talked about causality. So association between expression of a particular gene and a genomic variant is an eQTL. Most QTL studies are cis, that means within 1 to 2 megabytes of the transcription start site, so that's that beginning kind of location of the gene, as opposed to trans, which is more than that, or on another chromosome. This is starting to change, which is very exciting as computational methods and computing power gets better. Trans eQTLs is something that folks are able to do, and in fact, GTEx, the big RNA-seq consortium, actually has all of their eQTLs published, including the trans ones. And so if you're looking for tissues of interest and those kinds of things, that can be a really cool resource. So work in the biological area of interest is often required to determine mechanism of action. So we might go to model systems, which could mean cells in a dish. It could mean an animal model like a mouse or like a fruit fly. Often there's a lot of follow-up that goes into these just initial findings. So previously we had talked about DNA structure and organization. We hadn't talked about how this is all wrapped up. So this is hopefully going to transition us into a little bit of discussion of epigenetics, as I know that's also a sort of up-and-coming way to start looking at these data. So we talked about how everything's coiled again in chromatin, and we talked about how methyl groups can sometimes attach themselves to cytosines, and if you have a lot of these in one place, it leads to tighter coiling of DNA and less accessibility to the transcriptional machinery. So epigenetics is exactly what it sounds like. Epi on top of, genetics, the genome. So methyl groups bind to the cytosine bases, and these create CPG sites. And increased methylation decreases the binding of RNA polymerase, which we know we need for transcription, as we talked about yesterday. So this is a cartoon of what you might think about. So if we're all coiled up, RNA polymerase can't get in there, and then the gene is switched off. Silent. This is considered silent or condensed chromatin, and the histones also tend to be deacetylated in this case. So this can actually also go the other way. Acetyl groups can bind to the histone. So those are the proteins that the DNA is wrapped around. And when they're deacetylated, the chromatin becomes more densely packaged, which reduces transcription, which tends to travel with methylation. But if you acetylate the histones, the chromatin tends to be more open. And then more accessible to the RNA polymerase. And so there's this kind of balance where you have more acetylated histones, means open chromatin. It's available for the molecular machinery to go in and make mRNA, versus closed chromatin, where you have methylated cytosines, deacetylated histones, and closed chromatin, meaning that you don't have a whole lot of transcription. This process is dynamic. It changes over time, over development, and across cell type. So in very simple terms, you could think of this as on and off, but there is, of course, kind of a continuum between them. So there are some diseases associated with epigenetics. Prader-Willi syndrome is a mutation on chromosome 15, which ends up changing the methylation pattern and silencing the paternal copy of a region. This results in short stature, obesity, and has an OCD-like behavioral phenotype. It's located in this region, and then this is the area that ends up getting silenced. An interesting observation about this particular region is that if you have exactly the same mutation on chromosome 15, but in the maternal copy, you actually end up with Angelman syndrome, which results in microcephaly, severe intellectual and developmental disability, and this is thought to have to do with differences in methylation in fetal development. Yeah.
How does the cell know whether it's the maternal copy or the maternal copy?
That is a great question, and it's a little bit outside of my realm of expertise, but my if my memory serves from developmental biology, it has to do with how epigenetics is used in the developing embryo. And specifically, the one step of this that I remember quite vividly is that when an egg is fertilized, the paternal half of the genome is basically silenced. And so very early stages of development are carried out, and this is like initial cell divisions, like going from fertilized egg to something that has more than a couple cells, is done on the maternal genome alone. Yeah.
Is this something that's potentially reversible? Like when you, in a hypothetical world, imagine creating a drug that's going to open up that region, and would it even make a difference if you apply that kind of later in development?
Also outside my realm of expertise. I don't know of very many chemical structures that modulate epigenetics in a way that we understand. That doesn't mean there aren't any, that just means that I don't know of them. But certainly with this developmental stuff, it seems like it would have to happen as development is happening. And it's not intuitively obvious to me how you would fix it later. I hope that at least addresses your question.
Yeah.
These are pictures of children afflicted with each of these syndromes, and you can see that they're actually ending up with very different phenotypes despite having exactly the same mutation on chromosome 15. Yeah.
So would one theoretically use a QTA for the intensity of methylation as a given its need to establish causal relationships? So long as you know the organ type, for instance, whether it's a brain or lung cell, if you have that data, you can use that as a relationship.
Okay, so expression data is going to measure the mRNA directly, but epigenetics data is actually sequencing the cytosines on the DNA. So those would be orthogonal, both orthogonal data types to your SNP data and to each other. So it's very difficult to say a whole lot about methylation status from expression data and vice versa. But if you have them all kind of pointing in the same direction, that starts giving you a key or a clue into mechanism. Does that address your question?
Yeah, I think what I was referring to is for instance, if a given QTA shows that there is differential implication of a specific SNP, that is biologically important in terms of expression of a given gene. Is that?
I don't think I'm following your question.
So for instance, if there is a SNP, and there is a methylation profile that you do, and then you know that that specific SNP is differentially methylated for a given drug. So for instance, if you think of a smoker versus a non-smoker, if you have differentiated methylation intensities at that SNP, would that count for?
Okay, that would count as a piece of causal evidence. Certainly wouldn't get you all the way there. But just backing up a little bit, usually methylation data is not measured by a QTL. That's just sequenced directly. And there was a second piece. It's very unlikely that your SNP itself is going to be methylated because it's only the cytosines get methylated. So if you happen to have like a CT SNP, you could demonstrate that it couldn't be methylated because it's no longer a cytosine, but you'd probably want to look in the region more broadly than just at the specific SNP. But yeah, the logic that you walked me through is solid. Cool. Okay. So in this particular example, silencing of the paternal genome upon conception when the egg and sperm join has a totally different result from silencing or a deletion in the maternal genome. And this is to Dan's question, which is kind of the earliest stages of development are driven by the maternal genome alone, which might suggest that this is why the phenotypes are so drastically different. So another example of epigenetics that is not associated with disease is X inactivation in females. So the female karyotype is XX, males are XY. There's no difference in expression for most X genes between males and females. And you would think, how could that be? Since females have literally twice as many copies of the X chromosome genes as males. And the answer is that methylation is used to silence one of them. This is a randomly selected per cell X chromosome in females. So depending on which copy of the X chromosome is silenced, you can get different phenotypes if there are. So it's another cat example. Cats who we would call tortoiseshell cats have patches of orange and black, and the idea being that the inactive X is not being expressed for coat color, and the one that is active, those genes end up being expressed, and so this is why you get like bunches of both, is that like some of the X chromosomes have one X turned off, or some of the cells have one of the X's, one copy of X turned off, and then other cells don't.
Is it different that it comes in patches? Like if it was totally random and independent, right, you'd expect it to just be
right.
somewhat uniform.
That's a really good question, and I was looking for the answer to that. Perhaps Dan has the answer to that.
I thought it was patches because it happens at some stage where there's a limited number of cells, and then there are cell divisions that happen after that.
That sounds completely plausible.
Similar to the other cat example.
Yes, yeah, like the face, yeah, with the somatic mutation that happened early on in development. But they do seem more random in some patches than others, right? So this is just kind of to wrap up our thoughts about epigenetics. This is part of a dynamic system which gene expression is regulated. It's poorly understood systematically, but specific examples are decently well understood. And you can read papers and more about those specific examples. Epigenetic marks are regulated by DNA sequence and other factors. So proteins, but possibly other factors. And these things are all dynamically expressed in the cell. And so this gives us epigenetic marks that are regulating expression of the genes near them in real time. So this is getting into assaying epigenetics. So assays of epigenetic marks are emerging technologies and tools for follow-up of genetic results. This is a picture of the roadmap browser, which is a huge aggregation of epigenetic data, and all of this data is publicly available. An identification of an epigenetic correlation with a genetic variant is not usually enough to demonstrate causation, but it is certainly a nice piece of evidence to accumulate along the way. Often, actual lab work in the area of interest is also required to determine the mechanism of action completely. So, yeah.
Question on epigenetics. I've always been kind of skeptical because in my mind it makes much more sense to go and measure the resulting transcript and do RNA-seq. Is there any additional benefit to using epigenetics over transcriptomics if you're looking to find causal pathways?
Okay, so full disclosure, I'm definitely in the same camp. I work on mRNA data most days. So I would say that technology is more mature, which makes it often a better place to start. Epigenetics, I think, can be helpful if you're looking at something that is decreased in expression. Like if you want to look for that association with your SNP and expression, you understand that your SNP leads to a decrease in expression, and you want to see if that correlates with an epigenetic change. I could see how that could really strengthen an argument. But if you have to make an either-or decision, I think it would depend on the problem.
So does methylation change across the life course? also, for example,
Yes.
if I have like infant data versus like older people, would it, but so it changes, then would it change sort of universally across like CPG islets versus shores, or would the rate of change be different?
So the answer is, I think, yes to all. So certainly CPG content is going to influence how many genes you have in the region, and so there are some parts of the genome that stay bound up with methyl groups throughout the life course. There are going to be other places that stay open, say chunks of the genome encoding ribosomes or some very crucial cellular function. Like you can't have cellular life without ribosomes, and so, or we can't. And so that would stay open, and then there are other things that are more dynamic. Probably also unique to tissue. Does that help?
Yeah.
Okay.
Thank you.
So moving away from epigenetics, unless there are more questions about epigenetics. Yeah.
So like epigenetic changes also happen in vitro, right? Like
Mhm.
so there's a lead exposure in vitro and then compared to someone who tasted like an own line. So how do we compare like a person who has in vitro versus like someone who has like a half-life exposure?
I see. I don't have a good answer to that one. I think you'd probably want to understand the time course of the methylation of the regions that you're interested in. And again, this is a trade-off of doing things genome-wide, where it's really a lot to digest and understand, versus doing things in a very specific example, where like if there was a specific region of the genome that you were interested in, and you could find epigenomic data kind of throughout life, that might give you a better idea of what you would expect to change in background and what's in common due to the lead. But without that information, it's very difficult to tell which is which.
Also, just like since this definition starts in here, so do you think this can be used like a preclinical diagnosis in a symptom
What was the question?
Oh, the question was, can we use epigenetic data as preclinical diagnosis?
Even before like the symptoms start?
That's a really interesting idea. I'd have to think more about it, but there are certainly a growing population of researchers that are interested in looking at this kind of prodromal set of symptoms using a number of omics technology. So I might direct you towards that literature to think more about that.
We also have a whole epigenetics lecture next week too.
Yes.
So we'll be able to talk a bit more about that as well.
Sweet. Yeah, I don't know if anything that's an exact epigenomics example, but there are some examples in expression space. All right. Patrick, do you think it is better to stop now since we're running a little late for lunch?
Yeah, let's take a break now though.
Yeah, because